Hi everyone, today we're going over Chapter 4 of Biochemistry for the MCAT. Chapter 4.1 is about carbohydrate classification, and carbohydrates are basically carbon skeletons with a certain number of waters added. So for your general carbohydrate, this formula will be CnH2OM, where N and M are integers. And for monosaccharides, which is the most basic sugar unit, it consists of only one unit of sugar. This formula is CnH2ON. So if you see something like C6H2O6, you'll know that this is a monosaccharide that has 6 carbons in it. We can name carbohydrates based on their number of carbons. So a triose or a triose has 3 carbons, a tetrose has 4, a pentose has 5, and a hexose has 6. You rarely ever get above 6 carbons, but it's possible. The second fundamental difference between the different types of sugars is between an aldose and a ketose. So if you remember from organic chemistry, as these names suggest, aldoses will have aldehydes and ketoses will have ketones. So here I've drawn the most basic aldehyde and the most basic ketone. These are both trioses because they only have 3 carbons. So on the left we have glyceraldehyde, which, as the name suggests, has an aldehyde, which means that the carbonyl group will be at the very end of the molecule. On the other hand, dihydroxyacetone is a ketone, as the name suggests, so it must have a ketone group, which means that the carbonyl carbon is not at the very end of the molecule. And so the carbonyl carbon for an aldehyde is always known as C1. We can number the carbons as 1, 2, 3 from the top to the bottom, and by convention, we always place the carbonyl carbon at the very top when it's an aldehyde. It's not very clear in this picture, but when you have a ketose, the ketone carbon is generally the second carbon. So if you had a 6 carbon sugar, this carbonyl group will still be carbon number 2. You'll just have extra carbon groups in the middle here. These sugars can form what is called a glycosidic linkage, which means the linkage between multiple different units of sugar. When they link through glycosidic linkages, they will no longer be monosaccharides and will instead be disaccharides or polysaccharides. So this kind of linkage always happens at this carbon here, which is the carbonyl carbon. So for a ketose, this always happens at C2, and for an aldose, this always happens at C1. Something else you'll notice is that every other carbon has an OH group on it. And this is because if we look back at the name of a carbohydrate, it's a carbon with a bunch of waters attached. So it's natural that we should have OH groups as well as hydrogens attached. There are 4 monosaccharides that are so common that you're expected to remember them for the MCAT, and at the very least be able to tell them apart. So the first is fructose, and luckily enough, all of these 4 monosaccharides are hexoses, which means they have 6 carbons. Fructose is unique in the sense that it is the only ketose, and so you can always tell that one apart. Glucose, galactose, and mannose are all aldehydes, and they're all stereoisomers of each other. And you'll see here's the aldehyde group. There's actually a really easy way to remember these with your fingers. So first pretend like your thumb is the aldehyde or the carbonyl group, and also excuse my poor drawing skills. If you know that the only difference between these sugars is the positioning of the OH group, either on the right side or on the left side, you can use your fingers to represent each OH group. So for glucose, you'll stick your hand out and have your middle finger out, and this tells you the positioning of the one OH group that's to the left. And you remember this by saying F carbs. This is the hand gesture you give to carbs. Galactose has 2 OH groups facing the left, and you can think of this as your middle finger and your ring finger. So when I try to make this hand gesture, I can't do it, and I think that if you can, then you're probably not from this galaxy, and that's how I remember galactose. And lastly, for mannose, mannose also has 2 OH groups to the left, so this kind of makes a finger gun, and the saying to remember this is shoot the man. Next, we're going to talk about stereochemistry, which is about stereoisomers. And if you remember from organic chemistry, these are molecules that have the same connectivity in the same atoms but are different in space. So if you look at these 2 depictions of glyceraldehyde, you can see that they are the same in connectivity, however, their spatial orientation is different. So if you recall Fisher projections from chapter 1 and how to turn this Fisher projection into a version where you can see the stereochemistry a bit better, you can see that this glyceraldehyde is known as D and this is known as L, and how you tell the difference between these two is that the carbon that's second to the bottom will have the OH group on the left and D will have it on the right. A quick formula to calculate how many total stereoisomers there are with the same backbone is the formula 2 to the n. This is asked about quite frequently on the MCAT, where you're shown a sugar molecule and you're asked how many other stereoisomers there can be. And it's important to note that this N doesn't stand for the number of carbons, it stands for the number of chiral carbons. So if we look at glyceraldehyde, we can see that this top carbon is not a chiral carbon because it only has 3 groups attached. And this bottom carbon is also not a chiral carbon because it has 2 of the same group attached. It has 2 hydrogens. And so we only have 1 chiral carbon, and therefore 2 to the 1 is 2. And so we only have 2 possible stereoisomers, and these are the 2 enantiomers of glyceraldehyde. There are 3 rules for categorizing the stereoisomers of sugar molecules. The first 2 are for organic chemistry in general, and the third is exclusive to sugar molecules. So the first rule is that mirror images of the same sugar are known as enantiomers, and they're either D or L. The second rule is that sugars in the same family, which means that they have the same number of carbons and they're both aldoses or ketoses, are diastereomers of each other as long as they're not enantiomers, or if they're not the same. The third rule is that epimers are a subcategory of diastereomers, and they differ at exactly 1 chiral center. The exception to this, of course, is glyceraldehyde, because glyceraldehyde only differs at 1 chiral center, but these are mirror images to each other, so these are actually enantiomers. As an example, we're going to go through the different stereoisomers of aldotetrose. As review, the reason why this is called aldotetrose is because this is an aldose, since the carbonyl is at carbon number 1. And this is known as tetrose because it has 4 carbons. So first we can number these 1, 2, 3, and 4. And when we look at the relationship of 1 to 2, we can see that they differ at exactly 1 chiral center. And we can also see that these are not mirror images of each other, which means that 1 and 2 must be diastereomers of each other, and they're also epimers. And as a side note, the second to bottom carbons of both 1 and 2 have the OH group on the right side, and therefore these 2 sugars are both D. Looking at the relationship between 2 and 3, we can see that they differ at more than just 1 chiral carbon. They differ at both the chiral carbons, and you can also see that these are mirror images of each other, and therefore 1 and 2 are enantiomers of each other. Looking at the relationship between 3 and 4, we can see that they also differ at exactly 1 chiral carbon, and therefore 3 and 4 are both diastereomers and epimers. 3 and 4 both have the second to bottom chiral carbon OH group on the left side, so these are both L sugars. 1 and 4 are mirror images of each other, therefore 1 and 4 are enantiomers of each other. 2 and 4 are not mirror images, and 2 and 4 differ at exactly 1 chiral carbon, this carbon here, and so 2 and 4 are epimers, as well as diastereomers. And this relationship is also true for 1 and 3. Although every single diastereomer we've seen in this example are also epimers, this is not true in general. When you get to longer sugars, it's possible to differ at 2 chiral centers and be diastereomers but not epimers. Chapter 4.2 is about cyclic sugar molecules, and this is the mechanism for the cyclization of D-glucose. As you can see, the electrons on the oxygen of the OH group of the second to last carbon will attack the carbon of the carbonyl group, causing the electrons in that double bond to go back to the oxygen. And this is what forms a bond between the second to last carbon and the very top carbon, or the carbonyl carbon. And this carbonyl carbon is known as the anomeric carbon. You can see that this oxygen is now an OH group. As an aside, if you remember the functional groups from organic chemistry, this is an acetal and this is a hemiacetal. And so as you can see here, we formed a hemiacetal because here's the oxygen, here's the carbon, and here's the OH group. You can see it more clearly in the Haworth projection, where here you have your hemiacetal. This purple carbon here is still the purple carbon here, and the reason why this carbon is so important that it has its own name is that during this cyclization reaction, this OH group can actually point up or down, whereas all of the other OH groups have very predetermined up or down positions. For example, this OH group is always down, the next one is always up. You can see this if you turn this carbon skeleton sideways this way. And you can also draw your cycle sugar in the classic chair projection, where the anomeric carbon is also unknown as to whether the OH group is up or down. So I also drew here a ketal, because this ketal can also form a hemiketal if this here was a hydrogen. Aldoses, such as glucose, form hemiacetals, and ketoses will form hemiketals. Once a sugar is formed, it will either have the OH of the anomeric carbon pointing down or pointing up. If it's pointing down, it's known as an alpha sugar, and if it's pointing up, it's known as a beta sugar. And once this is set, it isn't forever. You can interconvert between an alpha sugar and a beta sugar, and this process is called mutarotation. And how mutarotation happens is that this bond here will break again, and the sugar will go back to being a linear sugar. And then the hemiacetal formation will happen again, except instead of attacking on the top, it would attack on the bottom, forming a beta sugar instead of an alpha. And so this is constantly happening in your body with all of your sugars. How I remember the difference between alpha and beta sugars is that if you look at the chair formation, you'll see that the alpha sugar has the OH group going axial, so alpha for axial, and the beta sugar has the OH group going equatorial, so beta for equatorial. The beta version of the sugar is slightly more stable, and so in your body about 66% of glucose or of most sugars will be in the beta formation. And a 6-member ring is known as a pyranose, while a 5-member ring is known as a furanose. So glucose most commonly forms a 6-membered pyranose. I don't think it's particularly important to know the mechanisms of how mutarotation happens or the exact mechanism of how sugar cyclization happens, but I do think it's really important on the MCAT to know where the sugars go once it becomes cyclized. So for example, you know that this is the anomeric carbon, and therefore it will appear here in the Haworth projection. You may be given a sugar molecule, and the answer choices will be 4 Haworth projections where you'll have to identify which one the correct one is. So it's important to know that if the OH group is on the right side, then it will face down, and if it's on the left side, then it will face up in the Haworth projection. You may also be asked what happens if a different oxygen attacks. So this is not the only oxygen that can attack the carbonyl carbon in terms of glucose. This is just the oxygen that would need to attack in order to form a 6-member ring. However, this OH group can also attack to form a cycle, but as you can see, the cycle will be 1 carbon smaller, and therefore it will only be a 5-member ring. That's how you would form the furanose version of glucose. I usually remember these by thinking that pyranose is kind of like fire and glucose is kind of like fuel for you, so therefore glucose likes to form a pyranose. For furanoses, I like to think about fructose, because as you can see, the carbonyl carbon for fructose is on the second to top carbon, and therefore when this OH group attacks, this will only form a 5-member ring, and so fructose can form a furanose. First off, we're going to talk about redox reactions of carbohydrates. I know a lot of people get very concerned when they hear the word redox, but redox is actually very simple in terms of organic chemistry or in biochemistry. So the most reduced that a hydrocarbon can be is its hydrocarbon form, and if you oxidize it one step further, it can be an alcohol, and if you oxidize it one step further than that, it can be an aldehyde, and the most oxidized form it can possibly be is a carboxylic acid. Oxidization can also take a different path where you're oxidized onto a different carbon, and this can form a ketone, but of course this ketone can never form a carboxylic acid because here you have a carbon which can never become an OH group. This is just a little organic chemistry recap. Like I said in chapter 1, I think that organic chemistry is a really good prerequisite for biochemistry. With this background, we can see that aldoses can be pretty easily oxidized. They can be oxidized by the OH group on their anomeric carbon to form a carbonyl instead. And this oxidation is called an oxidation to aldonic acids. because this in its linear form will form a carboxylic acid. Now, it's not very important to note that this is called an oxidation to aldonic acids, but what is important to know is that when you oxidize a sugar, the sugar acts as a reducing agent, which is why the sugar is called a reducing sugar. And when this does happen, you can see that you form a lactone, and a lactone is the organic chemistry word for a cyclic ester. This here is an ester group, and because carbohydrates are generally in their cyclic forms, we call these lactones. So some oxidizing agents that can make this happen are Tollen's reagent, which is more important in organic chemistry. And when Tollen's reagent is added to a reducing sugar, this forms a silver mirror. Benedict's reagent can also oxidize a reducing sugar, and this will form a red precipitate. So this is a really interesting question, which is, can ketoses oxidize? And the answer is yes, but they must first tautomerize to an aldose. The reason for this goes back to our linear forms of our aldose versus our ketose. So in our linear form of our aldose, you can see that the carbonyl is here at the very end, which means that our carbonyl can become oxidized to a carboxylic acid. However, when we look at our ketose, we can see that the carbonyl group occurs here in the middle, which means there's no way to turn this ketose into a carboxylic acid. So in order for this ketose to become oxidized, it must first tautomerize into an aldose. And it's not very important in biochemistry to know how this happens, but if you're asked in organic chemistry about this, you should be able to know how tautomerization works. Esterification is the process of an alcohol becoming an ester, and although this chapter describes it, I think it's very unimportant other than the fact that we know it can happen to carbohydrates and that the mechanism is pretty similar to that of phosphorylation. What's more important is glycoside formation, and here you have glucose, or this can happen to any other carbohydrate, but most importantly to glucose. And here you can see that you have the hemiacetal group described by the last video, and you can add an alcohol to this, and this can undergo Fischer esterification or just ester formation in general at the anomeric carbon, where here you have a new bond formed. This is the new ester bond, and in carbohydrates this is known as the glycosidic bond. So you can see that here previously we had a hemiacetal, and here we have a full acetal. But that's less important than the fact that this can be called a glycosidic bond, because I mentioned earlier that we add an alcohol to this, and you can see that carbohydrates have a lot of OH groups, and therefore this alcohol can be another carbohydrate. And when you have this alcohol as another carbohydrate, you can see that this bond could link to another carbohydrate to form a disaccharide or a polysaccharide. So that's why this is called a glycosidic bond, and once this bond forms, this is now known as a glycoside. So this bond most generally happens at the anomeric carbon, and if this happens to a furanose, this is now known as a furanoside, and it happens to a pyranose, it's now known as a pyranoside. That's not that important. Chapter 4.4 covers complex carbohydrates, and like we just described, we can form complex carbohydrates from monosaccharides. So first we can form a disaccharide, which is when you have 2 units of monosaccharides attached together by the glycosidic linkage, very important, this bond here. And here we have maltose, which is formed by 2 glucose molecules. So what's important about disaccharides and about glycosidic linkages in general is that they're named by the numbers that they're attached by. So here we have carbon number 1, which is the anomeric carbon, and here we have carbon number 4 because 1, 2, 3, 4, and so this is known as a 1,4 glycosidic linkage. You can also have this happen as a 1,6 glycosidic linkage, where 1, 2, 3, 4, 5, 6. So you can also have carbohydrates that are linked by this bond here. And these bonds can also be alpha or beta. The alpha version is important because this is a version that we can digest in our bodies, and the beta version is usually undigestible. Some common disaccharides are sucrose, lactose, and maltose, and these are the monosaccharides that they're formed by. These are the most common disaccharides. I don't know if they're worth memorizing. I've never been tested on these disaccharides, and I've never seen an AMC practice problem where you had to know what these disaccharides are. To clarify about alpha or beta linkages, that's in respect to the anomeric carbon on the left, where here, if it's axial, then it's alpha, and if it's equatorial, then it's beta. Before we talk about polysaccharides, I wanted to quickly talk about the difference between 1,4 linkages and 1,6 linkages. So here I've drawn a picture of purely 1,4 linkages. So if we pretend like this is a simplified glucose chain, that every single one of these points is actually a glucose molecule. So 1,4 linkage means that here's carbon number 1 and here's carbon number 4. And at carbon number 1, we can link more glucose molecules, and at carbon number 4, we can also link more glucose molecules. And so a 1,4 type of linkage forms a very linear sort of chain. If we have 1,4 and 1,6 linkages, on the other hand, here we have a 1,4 linkage, and if we had a 1,6 linkage, that means that at carbon number 6, which is the carbon here, we also can have a separate chain going off of it. So a mixed 1,4 and 1,6 linkage means that along with this linear type of chain, we also have separate chains coming off of carbon number 6. This is known as branching. Knowing this, we can talk about the 3 kinds of polysaccharides that are most relevant to biology and biochemistry. The first is cellulose, which is important because it forms most plant cell walls and plants in general, and cellulose is not digestible to the human body, and this is because it's mostly formed out of beta-D glucose linked by beta-1,4 glycosidic bonds, which, as we mentioned, the beta glycosidic bonds are not digestible by humans. So starch and glycogen are definitely digestible by humans. Starch is formed mostly of amylose and amylopectin. Amylose is glucose that's mostly linked by alpha 1,4 linkages, which means that amylose is mostly linear, while amylopectin is also mostly alpha 1,4 linkages, but it has also a whole bunch of alpha 1,6 branches. Glycogen is pretty similar to starch. It's pretty similar to amylopectin, but it has even more alpha 1,6 glycosidic bonds. In fact, glycogen has 1 in 10 alpha 1,6 bonds, while amylopectin has about 1 in 25 alpha 1,6 branches. And glycogen can be digested by glycogen phosphorylase, and glycogen can stimulate faster energy release. By the way, starch, which is amylose or amylopectin, can be digested by the different types of amylases. The reason why more branches can lead to faster energy release can also be explained by our picture of the 1,4 versus the 1,6 linkages. So if you only have a polysaccharide with 1,4 linkages, then you have an enzyme that can chew down your 1,4 linkage one by one, starting from the top all the way to the bottom one by one. However, if you have 1,6 linkages, you have enzymes that can chew down your 1,4 linkages one by one at each of the many branches. And in addition, you have enzymes that can cleave the 1,6 bonds. And so if you have more branches, that means you have more starting points and more places for your enzymes to be chewing away at all at once. Thank you so much for watching. I recorded this chapter in 2 parts and then I realized that it could fit in one video, so thank you so much for bearing with me and I hope you enjoyed this.